We see that in microarray, the process of hybridization is important. So we have DNA to DNA hybridization or DNA to RNA. So whatever the case may be, let's see how we design these arrays. So the DNA is deposited on the slide by different methods. One is printing. So with what is an inkjet printer is used. We do have a spotting method in which we have robots which are having set of fine tip pins. So with the help of those robots, we can put or transfer that those cDNAs or DNAs into those slides. Now we call this as spotted array, this technique. So here we see the inkjet. We have Canon technology, HP Canon technology. So we see those DNA are spotted or shooted through these pins, through these kind of those, they are printed on the slide. In spotting, the DNA is attached to a solid spot, glass, plastic or nylon. RNA is labeled, so which is usually indirectly labeled. So in this way, we when we hybridize them, we see that the this bound DNA is the probe, which is actually present on the slide, whereas the labeled RNA is the target. So probe, target. The probe is present on the slide, so which is actually transferred with the help of those robots or with the help of those printers. So here we see the spotting printer. So we see there is a robot or there is a machine, and we see down there are the slides, and in this we have those large number of pins which are actually transferring this DNA to those slides, and then that DNA has capacity to get attached to those glass present in that slide. This commercial spotter, we see that this is a picture of this commercial spotter. So we have a large number of slides which are present, and then the spotter is actually, with the help of those pins, it's transferring into each of those slides those probe cDNAs. In Informatics gene chips, we have oligonucleotides which are synthesized on silicon chips. One base is transferred at a time, and it uses the process of photolithography, which is actually developed for printing those computer circuits. And oligonucleotides for each genes are selected by different computer programs. So depending upon, a good oligo should be unique in that particular genome, or it should be unique in capturing those typical genes. And then if we do not have those unique, these probes are not unique, so then there will be, we can have the cross hybridization. So we can it will be hard for us to distinguish between 2 or 3 genes. So they may go and their messenger RNA can go and get attached to the same spots. And these also should be non-overlapping, these oligonucleotides. So in Informatics, each gene is represented by a probe set, which is consisting of 12 to 20 probes. Each of them is 25 nucleotide length. Each probe has a corresponding mismatch probe with a single base difference at the 13 nucleotide. Labeled RNA is hybridized to the array, and abundance is calculated using hybridization for the entire probe set, correcting the mismatch probes, which actually indicate the non-specific effect. So we do have a mismatch over there, so that tells us if we can have a relative abundance between the match pairs versus the mismatched pairs. So in this way we can correct for those mismatching probes. So here is the chip that looks like this, and here we can have those probes which are present in 2 layers. So here we see in this diagram a little detail about how these probes are prepared. So we have 12 to 20 per gene. These probes are prepared. They may be covering the whole genes or some important, mostly it takes the important regions of those genes. So we take that messenger RNA, and for that down below you can see we do have a match oligo, this one, and then we have a mismatch where we have just one nucleotide is mismatching. So they are arranged in actually 2 layers. If you look down below, so we can have a fluorescent intensity image which is coming from them. So we have those probe pairs. So on the top layer, the white ones where we have those perfectly matched probe cells, and then in the bottom layer we have the mismatched probe cells. So in this way it's kind of helping normalization of this data or correcting for those mismatched pairs. Unlike those glass-based arrays, these Affymetrix chips, we can analyze only one sample, so we cannot actually compare two samples. But obviously, for one sample, you can get the results, and you can take another sample, run it on a chip, and then you can compare the results with one another. So no direct comparison, guys. But the good point is that this procedure is a standardized protocol. Since they keep on improving their technologies, they keep on doing their R&Ds, and this Affymetrix has been a good company which has been the prominent among those microarray kind of technologies. So we can have those standardized production of those microarrays, so that can give good reproducibility. So you can have these results reproduced or replicated. It's hard to replicate the results with the microarrays as far as the glass arrays are concerned. You cannot get the same exact situation, so you can have variability among your experiments. So, but this Affymetrix claims to control that variability, so we can reproduce our results. Another thing is that limited amount of this probe sequence that can also be complicated, or that can also be a problematic. So you can miss alternative splices, it can be biased towards the ends, but well, that depends upon how good your genome annotations are. Let's talk about the attachment of those sequences to the slides. So one of them is they are attached covalently. So we can have those aliphatic amine groups, and they are added to the 5' end of those probes, and then those probes are attached to the glass from those 5' ends using these groups. In non-covalent attachments, we can have electrostatic attraction between amine and phosphate on the probe's backbone. A CDNA is attached to the glass by the backbone, and this happens only in cDNA microarrays. Oligonucleotides are short for this kind of interaction, so if you want to use a backbone, you must have a large or a long backbone. In another scenario where we are using those in situ synthesized oligonucleotide arrays, for example, we talk about Affymetrix. So here the nucleotides are added one by one as far as those probes are concerned. So we have base by base addition on the array surface. There is a covalent reaction between the 5' hydroxyl and phosphate groups, and we add those protective groups on 5' position to prevent the multiple addition. So you add one nucleotide at a time in order to prepare that probe in a smooth fashion. The protective group is then converted into hydroxyl group. This is how it happens. So you have those solid spot, you have a base which is added, and then you add the next base, nucleotide is added, and then the OH of this base is actually protected here. So you see that PG, and then you can use acid or light to control this process and make it deprotected, and then you can continue accepting. But at that time, at only one nucleotide is added at a time. As far as the labeling is concerned, we use biotin that is there in the oligonucleotides, the Affymetrices, and we see that this biotin is then attached to streptavidin, which is attached to phycoerythrin, which is a colored pigment, so that then can be used to give a color display, or with that colors we can get the idea of those relative expressions. Whereas the fluorescent dyes which are used in these microarrays, which are glass-based macroarrays, we have CY3, that is it is excited by laser at a wavelength of 532 nanometers, and it gives a fluorescence green color. Sometimes it's also called as dye 532. CY5 is also known as 635, so where it is excited at a wavelength of 635 nanometers, so it fluoresces red. So in microarrays, we do have complementary strands of DNA and RNA. They hybridize with one another.